IISc Banglore's Dr. Aloke Kumar and his students are sending an experiment to space that will grow and multiply bacteria. They even received an award from ISRO for exactly this design. This experiment is important if you want to build structures on the moon. And guess what? This experiment or this video will be a first look into a Gaganyaan experiment. For those who don't know, Gaganyaan is India's mission where they're sending astronauts, Indian astronauts to space on an Indian rocket. This video also gives us a rare look into how Indian universities think about a space experiment and how they approach it. So a lot of new stuff. These are the things we'll be mainly going in this video, from bacterial multiplication to pump systems to how bacteria is counted, gravity effects on bacteria, fail-proof design, structural design, electronics, and even how vibration and waterproofing is done. It's very interesting. Let's first give you some background. 3 years ago, with Dr. Alok Kumar, I made a video on his moon bricks, where they took some lunar soil simulant, which is like moon soil, mixed it with bacteria, some food for the bacteria in a bioreactor and calcified it, basically made it hard. This then they could machine to make proper bricks out of, right? So you could use this to make structural elements on the moon. Now, in a follow-up video, they also made some Mars bricks, which also I covered in a video. Now the thing is, these experiments were done on Earth, right, in gravity. Suppose they want to do this experiment on the moon in future, suppose in a Chandrayaan mission maybe, they have to understand what this bacteria does in space. Maybe it doesn't even grow or doesn't even multiply. This is something that aim to validate with MANAS, which stands for Microbial Analysis in Space. We'll understand this in detail. Let's start this video. This is Dr. Alok's student Santosh who will be explaining this. The approach I'm taking to this video is he'll explain something, I'll stop the video, then I'll explain you and give you more context. While this conversation with him happened mainly in English, so this video is fully in English, but you can also switch the language to Hindi commentary, where this commentary will switch to Hindi. First, we will understand cassettes, which are the containers where this bacterial multiplication happens.
So, one cassette will be holding multiple experiments, here 4, and we have a set of
You can see something like S. pasteur written in this diagram. That is the name of the bacteria. Below is the nutrient broth, which is the food of the bacteria. Remember I told you about the whole bacteria growth thing. So there are bacterial spores which are like seeds. You give it food, it grows, it multiplies. Now the thing is, you need a space to do this, right? And this is what cassettes do. Each cassette has 4 wells. Each well you can grow this bacteria. Now there is a problem. How will you control the growth of this bacteria? What if this bacteria grows while on the pad or even before the rocket launches? So it's important that you build a system which allows you to grow the bacteria only when you command it to.
To send the, as I was talking to you about the experiment not starting at the right time. So we have bacterial spores in certain solution which ensures that they do not grow before a certain point. And then we have the food separate. What we do is we feed the food at the right time through a complicated network of fluid circuitry. But the idea is that.
So some things will of course remain secret. But one thing I was curious about is when you hear cassettes, you feels like you can stack them. Like, can you have one stacked second cassette and the third cassette on top? So I asked Alok sir about it, and this was his answer.
So, I mean, if you have such a cassette, does it mean you can add multiple? Like those cassettes are stackable, or is this modular in sense?
I think my professor will be able to answer this better, but I think that is the idea.
Cassettes की बात करते हैं, तो I would assume multiple or.
इसीलिए cassettes है, क्योंकि what we want is easy scalability. अभी तो we have been given a certain dimension, size की भी constraints में काम करना है. But इसको अगर scalable करना है, that's why the cassettes are there. You can add multiple cassettes, you can change the sensor. और हम लोग जो बना रहे हैं वो सिर्फ अपने लिए नहीं बना रहे हैं. We are also making it for any other biologist जो बाद में space experiments करना चाहता है. They can take our system और इसको replace it with their sensor if they want and add any more small changes. So the entire design is extremely modular in nature. ताकि अलग-अलग portions swap हो सके, more cassettes add हो सके, sensors change हो सके. Everything is easily swappable, easily within ports.
Okay, but will your pump scale with it if multiple cassettes are there?
Yeah, it will.
It will scale. It depends. It actually depends on a little bit of कि कितना volume चाहिए. But ये अभी जो fluidic network है इसमें it is not using the pump to the full capacity at all. So we can easily add more cassettes. So it's a little bit dependent on the exact experimentation. Hopefully, अगर volume बहुत ज़्यादा ऊपर नहीं जाता, then it is not a problem.
Okay. So like I said, there are 4 wells. There are pumps which pump food into these wells, and this will be only pumped when they are given command to pump, and hence bacteria shouldn't grow if they are not fed food.
Right to say that this is one of a kind MilliFluidic experiment. Like other people have microfluidic experiments, the volume is like 100 microliters. Each chamber here holds like 1 milliliter. So that's 10 times one chamber there. So we already have that. And
so when you say 4 wells, 4 different bacteria or 4 different
same
sections?
bacteria, but to bring in redundancy, also one will be control. So that will be my reference line. And the other 3 experiments will get me, so will be 3 different experiments in triplicates.
Okay.
So, and these are independent individual experiments. So one is not related to another. So that is what we are maintaining here.
So this is important that out of 4 wells, each well has 1 ml, but one well is controlled, meaning there won't be any bacterial spores in this, only food will be pumped and then it will be used like a baseline to compare to others. Also, we need to see that a similar experiment is running on ground at the same time, so we get better comparison data. And when I said bacteria will be counted and grown, how will you count this? And here comes the magic of LEDs.
Imagine these are my experiments.
Right.
And the simplest experiment that we can do is understanding the growth dynamics of a bacterial species, which we do through understanding its optical density curve.
Okay.
So,
understanding its
optical density.
As in, you shine a light and see how many are there.
Shine a light and see how many are there. Exactly. So, in a way, these curves are very characteristics of each species. So, for example, I go to the results first to explain.
Right.
So, there's a lag phase. There's a log phase, then it peaks here. Then what happens is, due to resource crunch and many other issues, it starts dying.
Okay.
And this is called the stationary phase. So these are plotted in logarithmic scales. So this is with respect to the initial reading, and this is a standard procedure that we biologists do for this process.
If you did not understand, let me give a simpler explanation. Imagine there is LED on one side and there is a receiver on the other. Ideally, if the LED is on, you will receive the entire light on the receiver. But if something starts blocking it, you will receive less. And if even more molecules block it, you will receive even less. This is a relation you can draw for bacteria as well in a liquid, and hence kind of predict how much bacteria is there, right, through optical density. Look at this graph. From what I understand, initially you have a certain amount of bacteria. This bacteria will grow and multiply as you give it food, but eventually it will reach a point where the food has gotten over and there is not enough resources to survive, so bacteria will start dying. But this graph will also have to be validated in a microgravity environment if it's similar. But what kind of LEDs can you use? Any LEDs?
Okay, why 600 nanometer, and does it affect the bacteria? Does it change its behavior?
Yeah, so the length to which it goes to, the bacterial species goes to, maximum scattering is at 600 nanometers.
Okay.
So for this species, so it goes to around 10 micrometers in length, as in it's a cylindrical this thing, and 6 to 10 micrometers is average length, and 10 micrometers at max. And this is the wavelength for maximum scattering.
I was curious, what will no gravity do to a bacteria? So I asked.
So what are you expecting its output to be? Like you feed a certain amount of suppose X bacteria. After the experiment, what should be the?
So even while doing it in lab, because this is fully sealed, right? There is no aeration column.
Correct.
In general, we have aeration column.
Correct.
We put it in incubators.
Right.
Even here, we have seen some sort of stunted behavior. However, the growth curve has been followed, but the maximum range has reduced.
Okay.
So we have seen that. What we are expecting is a delay in the curve. That's an hypothesis. What we are expecting is a delay in the curve and also much stunted growth. So have one replica of the model on ground, one replica there, and then compare the.
Okay.
So the whole idea is for the microgravity.
Okay. So far you don't know how. Do any other experiments, similar experiments of how microgravity affected bacterial growth?
For this case or any other?
Any other, like
any species.
So it is known that, for example, pathogenic species become more pathogenic in microgravity, and behavior of cells definitely changes. A bacteria is not a pathogenic bacteria. The A bacteria purpose is very different, right? The space breaks, and this is a good bacteria that exists in the soil. This we don't necessarily know how much, what is the effect of microgravity there, and what is not. So that's why it's an interesting question to look at.
It will be interesting to see the results of these experiments. But let's come to Manas and how does it look, what does it do, and what are its
That's
components.
exactly what we have avoided here. So this is just the system of cassette and different how each chamber is connected to the other. The main focus is on developing a complete system which inculcates all of these. So the first one is a PSU unit, power supply unit, and this is connected to the main bus line in the spacecraft. However, this so they have unregulated DC supply, and we need specific voltages. Otherwise, the data correlation will be fully wrong. So we have a series of buck converters and voltage regulators which ensure that the correct logic levels reach each point. Then, as I explained, there are valves and pumps which ensure that the liquid reaches at the right time. So for that, we have a fluid circuit control, which controls the which ensures that once it starts, once the experiment starts, I'll come to that how it starts. Once the experiment starts, all of these are diagnosed and all of the mechanical action is done.
Some might have found this overwhelming, but basically, see, there are some circuits that trigger the experiment and collect data from the experiment. There are some pumps, and then there is a cassette area where the experiment is happening. Now, before coming to the electronics and vibration and all of that, let's ask Aloke sir about this Gaganyaan thing, and is it actually flying on Gaganyaan?
ठीक है. Gaganyaan official है? Can we say that?
Selected for G2 at the moment. The experiment is selected for G2, provided it goes through all the qualifications.
Oh, qualifications like vibration
tests. So this has to be reviewed by an ISRO committee. The process it has already started. So now it has, at some point it will have to undergo all the tests.
Now I know a lot of people will ask me, when is Gaganyaan G2 flying? Look, these dates are never fixed, right? And especially for such a human critical mission, things will always change. G1 itself is scheduled for somewhere in 2025, maybe Q1, Q2, Q3, and after that, basis of that data collected, they'll decide G2 date. That might take another year or two. So this situation is always fluid. Maybe these people get another opportunity on another mission to fly this experiment. So these things keep happening. And before we discuss ISRO requirements for vibration and all of that, let's talk about the electronics and everything that goes into communicating with the Gaganyaan capsule and how it interfaces with it.
This thing. So this is our CDN unit, so control and data handling unit. So basically, this is connected to the external computer. So
to the spacecraft.
So because there is a trigger involved, because there are multiple payloads and ours is not the only one. So we send a signal via telemetry from the ground station, and this payload gets the go-ahead signal for starting the experiment. So it connects with everything through an internal I2C bus, and it's and it ensures that all the diagnostics and all the running are completely controlled. So the experiment is hosted here. We are continuously getting data. We have onboard data acquisition system as well, but we are also sending in data through telemetry to the ground.
This will be inside the capsule or on the service module outside? How is this?
It will be inside the capsule, right?
Inside
the capsule, right?
So its temperature and all will be controlled by the cabin?
The temperature needs to be controlled because these species grow at 37 degrees Celsius, optimum.
So, but you are controlling the temperature or the cabin's room temperature?
The cabin temperature works. So the idea is not to go beyond a certain temperature range, like below 5 or something, which will cause a lot of issues. But as in it will stop growing. But a certain range, the room temperature range, which is from 22 to 40 degrees Celsius, it works completely fine for us.
So you did not have to implement heating, cooling and thermal design and all.
So the whole idea is that power budget itself is 10 watts. 10 watts at the maximum, and nominal voltage is 1 watt. So so that's, and the volume, if you see, is 140 cross 140 cross 300 millimeters. So that's less than a A4 size sheet.
Right.
So the idea is to pack it as small as possible and ensure that it eats up less current and is as autonomous as possible. So the only thing we are doing is sending a trigger to the CNDH unit that to ensure that there are steps followed, as in the protocols are followed. This is a mock-up of a laboratory model that we are using. It doesn't have all the components. So this is a bigger version because for ease of use, we have made it bigger. But the original size, the payload that's going to go is 14 cross 14 cross 330 centimeters.
Okay, that is quite small.
Yeah, and this is also 330. This is 190 cross 150 cross 330. This is like 40 mm bigger than the actual dimension. That's
Right.
Now it's very nice for them that they do not have to worry about the thermals, right? Because imagine if this experiment was outside the capsule, they would have to do thermal design and thermal engineering and maintain the temperature with heaters, etc. It will be a lot of work for them. Since this is inside and the capsule has atmosphere, it has air, right? Since it's meant for humans, this will automatically maintain the temperature based on cabin temperature. The one thing with this experiment is that you only get one chance, right? It's very expensive to fly to space. So they have to make sure that this experiment does not fail. And if one component does fail, just in case, there is some backup, some redundant system that can take over. So let's talk about
No,
it.
as in, I think you have two pumps, right?
Yeah.
So they are redundant pumps, or
Yeah,
they
for
are-
redundant. For redundancy only, there are 2 pumps and there are multiple. So there is a whole diagnostic system. That's
So even from your power supply, it's redundant. The redundancy is there from the power supply.
yeah,
So you have basically like two systems running separately.
Yeah. So the idea is, and there are many other diagnostics as well that so we are closely monitoring each of the systems. For example, once the roll of pumps is done, it will be completely switched off. So that's why it's 10 watts for the peak thing, and then it will shut off, and no need of pumps and any other stuff. The only thing that will be happening is data collection here.
Right.
Now this is nice, right? 10 watts peak is nothing. It's like a LED bulb or some tubelight. It's not a lot of power they are using. Now let's look at how this module communicates with the Gaganyaan capsule, the electrical connectors.
So as you can see, this is one module. So if you see, there are 4 buck converters here. So any unregulated supply from the range 24 volts to 48 volts, that's what they have generally.
Right.
So that goes to 4 stations. One goes to the main controller, 5 volts. One goes to the small controller that's here at this circuit.
Okay.
And 2 go to the, one goes to the valves and one goes to the pump. So here you can see a series of MOSFETs here. So what they do is they assist in the switching of this. So there's a microcontroller here in this board as well.
Right.
It controls the switching of each of the valves, and then there is a H-bridge circuit that we have for controlling the pumps. So it's a dual H-bridge circuit. The basic idea is it's like a switching algorithm. So you send in particular signal, it works one way, and you send in a different signal, it will work the other way. So that's basically I'm simplifying the H-bridge.
Tell me, this look like VGA. Why do this look like VGA ports?
So this is a D-sub, standard D-sub. So this connects to the external interface.
Right.
So this is our power interface. Similarly, we have one in the main controller here.
Right.
So this is the communication port.
Right.
So we are communicating via RS-485 right now to the external computer. So there's a RS-485 line going here, and there's a power line coming in here. All these are, when you are asking about each module having interface, so all these are interface lines only. So each
module have a similar port?
Yeah. So each of them has, if you can see, each of them has. Even this module has one at the bottom of it. So what happens is, so you connect it to each module, and that ensures that there is no this thing. So it's completely modular.
Right.
Yeah.
Also thought it will be a good idea to ask Aloke sir again about what's up with the moon bricks and if there's a relation in this mission to it.
Right. So this will define like your plans of how bricks might
this
be.
is the first stepping stone there. First to send experiments, bacterial samples stored in a fully contained chamber or in an automated experiment. This is the first step towards it.
Right.
Because obviously for the bricks we'll have to build a much more sophisticated version of
Correct.
So take one step at a time. If this is validated, then we'll go to the next step.
Right.
I know many might say that this looks simple or something like that, but in reality it's very complicated because first of all you have to make a prototype, then you have to make the qualification model, then the flight model, and all of this have to work perfectly to convince ISRO to put it on there. Then there is another problem that the rocket is very aggressive. It looks smooth from outside, but it vibrates a lot. So every satellite has to undergo a vibration test along all of the axes X, Y, Z Now the problem is, since there is liquid in this experiment, it complicates this whole vibration testing and everything, right? Because you are in a capsule that is electrical connectors. So I was asking them about this vibration
And do
test.
you have additional issues like वो leak नहीं होना चाहिए या
all those have been already taken into consideration. मतलब जो leakage वगैरह, because this is completely sealed. Even the D-subs and all the interfaces, look, the actual D-sub, the mill grade ones have a sealing here. So वो सब हम कर रहे हैं, and the volume we are operating is like 5 ml or something.
अगर वाइब्रेशन टेस्ट पे लीक करें तो ये potentially उतना problematic नहीं है, but leak is, we have to show that it is leak. That is obvious.
And like since this is first time the ISRO is kind of flying a biology, I think, I'm not sure. तो क्या नए protocols और
नए protocols इसमें अभी तो, other than being leak proof and hermetic sealing requirement, there is nothing else. The biology, the bacteria is a good bacteria. It's a friendly bacteria, so that there's no issue like that.
Okay. Is the water conductible when bacteria is mixed in it?
A little bit, but the amounts are very too low.
Okay.
So, but still, it has to hold. All the connections have to hold, and no leakage should happen when it is undergoing, let's say, a vibration test.
Okay, vibration test are fine. What G-force test होगा?
हाँ, 18G.
18G pulse होना था versus whatever is normal.
Yeah,
like
obviously there's a factor of
access,
safety. There's a factor of safety, not just in terms of stresses, but even deformations में कि what's the maximum bending hold. So because it's random vibration and the bottom plate is fixed,
correct.
सबसे the maximum vibrations will be at the topmost. The maximum deformation will be at the topmost place, which comes around to 0.5 microns or something. Okay. So that is how you qualify that this is good. The maximum, absolute maximum deformation it can go through, the top plate that goes through is 0.5 micron.
So I hope you got some interesting insights from this video. On paper, making a prototype is easy. You can plug a Raspberry Pi, some sensors and whatnot and get it working. But making something for production that should always work, never fail, is a completely different ball game. So it takes years to make something for space, right? Because more than working, never failing is important, and these two philosophies are completely different. Thank you to all members who enable me to do this, to travel, because these things are expensive. Thank you so much.
जयम शर्मा, रोहित सुवर्णा, उटल चैतन्य, कार्तिकेय देवाल, विशाल भारत, कृपल पटेल, शौनक पेरनी, कुमार अखिल, चंद्रबदन सिंह, श्रीकांत कापड़ी, मुंड्र, विशेष वत्सल, व्हाट्स न्यू, विद्यासागर, रौनक. थैंक यू सबको.